but of course it varies from woman to woman. It also varies in what she does. If she's very active physically, sometimes that cycle may be altered, especially if she's very physically active. And in female athletes, particularly of Olympic caliber, that cycle, menstrual cycle, may just stop altogether during her her activity, because you need a certain amount of fat in the body. And if she's very lean and thin and trim, okay, there may not be enough fat because the fat will store estrogen, basically to support a pregnancy. So for that reason, it can actually shut down or become very irregular and then shut down. Okay, but on average, menstrual cycle in human females is roughly a monthly cycle, about 28 days. Okay. Now, we only see the menstrual cycle in primates, which are humans, apes, and monkeys. You know, there's always a lot of people who say we have any relationship to monkeys. We do, okay? It's not to say we descended from monkeys. No, we did not, but we have common ancestors. The point is, it's only those higher mammals, primates, that actually have a menstrual cycle.
What is it that the dogs go through?
They have what's called an estrous cycle. That's true of most mammals.
Okay.
And in that case, and it's like dogs, cats, many domestic animals, what happens? The female goes into heat. That's when she is basically ovulating. That's when she's most likely to produce a fertilized egg, okay?
And run off.
But it's only those times that the female is receptive to the male. And that's why if you got a female cat and she's not been spayed, she starts acting really strange. Okay, and they got the tomcats.
Even if they are spayed, they still.
Yeah, and then the tomcats start coming around because they get her scent. Okay, that's what attracts them. It's the hormones. Okay, now that's not the case in animals with a menstrual cycle, which include human females, who theoretically are receptive to male anytime. Now, obviously, that is up to the woman. Okay, how receptive or unreceptive she is, but there's not. She doesn't go into heat. Sometimes people say, "Well, she's in heat." No. Okay? That's just derogatory, saying, well, you know, she's very sexually active or whatever. Okay, but anyway, so we're going to say a 28-day cycle on average, and the first day of the cycle is the first day of menstrual flow. That's day one. It all starts with a hormone released by the hypothalamus, and that's the gonadotropin releasing hormone, GnRH. And what it does, as the name tells you, it stimulates the release of the gonadotropins, which you already know, you just don't know them by that name. That's FSH, follicle stimulating hormone, and follicle stimulating hormone and luteinizing hormone. Those are the gonadotropins because they affect the reproductive organs. All right? So, what this does is stimulate, okay, the release of FSH and LH. FSH, as you know, stimulates follicle development in the ovaries of the female. As those follicles develop, they release estrogens, female sex hormones, and this stimulates the growth of the uterine lining, the endometrium. So it's basically preparing the woman's body for pregnancy. Because the endometrium, the uterine lining, is where an embryo will implant if fertilization occurs, and that's essential, that implantation and the maintenance of the endometrium to keep the pregnancy going. If that sloughs off, you've got a miscarriage or spontaneous abortion. That's very important. All right. In human females, unless she's under the influence of a fertility drug, only one follicle usually continues to develop to maturity. For some reason, the others, which have started development, shut down, and those degenerate. They're called atretic follicles. But this one that continues to grow just prior to mid-cycle, and again, we're using it at 28-day cycle. This would be about day 13, and again, day 1 you can see is the first day of menstruation. All right, estrogen levels in the blood now peak, and this stimulates the surge in the release of LH and a smaller surge in the release of FSH. This is actually a positive feedback mechanism. A&P1, I'm sure you talked about feedback mechanisms. Probably negative feedback, which happens all the time to keep regulating things. Positive feedback only occurs periodically and then only for short periods of time. So this is a positive feedback mechanism. So it doesn't occur a lot, okay, roughly in a monthly cycle here, and doesn't last for very long. But it's thought that this surge in LH level, okay, is what triggers ovulation. And that's release from the mature follicle, it's a Graafian follicle, of that egg. Now the egg in this stage, called a secondary oocyte, is not mature. And it won't mature until what happens? What
It's-
stimulates the egg to complete meiosis II and become a mature egg or ovum?
The sperm.
In other words, it's got to be fertilized. The sperm has to physically gain entrance into the egg cell cytoplasm. Okay? Did I write on the board what the sperm has to penetrate?
The 2 layers.
Yes. Anybody remember what they were? Cells around the egg are actually cells of the follicle in which the egg develops, called the chromoidiota. Okay? That's one of the barriers the egg has to, the sperm has to penetrate to get into the egg cell cytoplasm. The other is actually the membrane, the external, the egg cell membrane. This is called the zona lucida. Okay. And that's why the sperm cell has to have digestive enzymes and literally digest its way through these cells and the membrane to actually physically get into the cytoplasm of the egg. And that's what stimulates the egg then to finish meiosis II, become a mature ovum, and then the nuclei come together. And you've taken 2 haploid cells, the sperm cell and the egg cell, and now you have a diploid, zygote, or fertilized egg. Okay? And that's why your gametes are haploid. Otherwise, every time fertilization occurred, you'd be doubling the chromosome number. And pretty soon the cell would be overwhelmed with chromosomes. And people, well, that's great, extra chromosomes. No, it's not great. 1 extra chromosome, okay, 23
syndrome.
or 21 actually, trisomy 21 is Down syndrome. And there's some that even more severe. Edwards syndrome and Patau syndrome and so on. 1 extra chromosome. So you want the exact number. All right, anyway, that follicle then, okay, releases the egg, and after it does, at ovulation, that mature follicle, which is ruptured now, under the influence of hormones, develops into the corpus luteum. That's the thing you can see on the ovary model. Here's the Graafian follicle, okay, got a big cavity here, fluid filled, and here is the secondary oocyte, the egg cells being released, and the cells around it, that'd be the chromoidiota, that stay with it. They're actually cells of the follicle that remain with the egg, even after ovulation. So what happens then, after this develops into this, which is the corpus luteum, yellow body in Latin. And this is a major female sex hormone producing structure. It continues to produce estrogens like the developing follicles did, plus a lot of another hormone called progesterone. Okay? The progesterone is essential because it maintains the endometrium to keep it viable. So if you got a fertilized egg, it can implant in the endometrium and continue development. Okay? All right. Now, though, high levels of the estrogen and progesterone, because remember they've been produced by the developing follicles and the corpus luteum, they inhibit further secretion of FSH and LH. And with that drop in LH is maintaining this corpus luteum. You get the corpus luteum now degenerating. As it degenerates, of course, the hormones they produce fall off, especially the progesterone. Progesterone is maintaining the endometrium, so now the endometrium degenerates, sloughs off, and you got the menstrual flow.
Yep.
And that makes up most of the flow, is that degeneration of the endometrium. Now, once that happens, then you get a drop in the blood levels of estrogen and progesterone, because now the corpus luteum is actually shriveled up, become the corpus albicans. So now FSH and LH secretion resume, and the cycle repeats itself. That's what usually happens each month, as long as the egg is not pregnant. But if the egg is fertilized, and where does fertilization occur in
In
humans?
the fallopian tubes.
Yes, and usually the upper third, closer to the ovary than to the uterus. So that's why the sperm has to be modal. It's got to swim. It's got to go from the vagina up the uterine cavity and then up the fallopian tube. And again, which one? It's often one or the other. The egg is in. That's why it takes a lot of sperm for the male to be considered functionally.
Adequate.
Yes. And typically, in a normal healthy male, there's something like 20 million sperm cells in that ejaculate. 20 million, if you can imagine that. Most of them are never going to make it. They're going to die en route, okay? And the one that actually does make it up there to the egg usually needs some help from his buddies, because you need enough of those digestive hormones to get through, at least one of them to get through. and usually only one does. Okay, so what happens then? With fertilization, embryo begins to develop, and cells around that developing embryo secrete HCG, human chorionic gonadotropin. And this is very important because this is what maintains that corpus luteum, which continues to produce progesterone, which in turn maintains the uterine lining, the endometrium, to keep that pregnancy going, okay? And if a woman is pregnant, this usually is maintained for about the first trimester, the first 3 months of pregnancy. Yes?
So that's what can cause a
Miscarriage?
yeah, if they don't produce enough of the HCG.
Yeah, if something happens and basically this is shuts down, okay, not produce enough hormone, progesterone, then the uterine lining will slough off, stratiform, and it takes the embryo with it. Okay. Now sometimes it's other things like trauma, or maybe disease that can also occur too. Okay.
Gotcha.
All right, anyway, HCG, so actually two hormones that maintain the corpus luteum. Initially it's LH, but that's going to fall off, okay, because the high levels of the estrogen and progesterone, okay? And then with pregnancy, it's going to be HCG, okay? And again, that's what you're testing for, because it builds up to such high levels in a pregnant woman, it spills over in the urine, and you can test with usually a dipstick. You know? And often it's sensitive enough, you can actually detect it before the woman realizes she's missed a period. Even after a couple of weeks. It's often sensitive enough to pick up after 3 weeks.
LH is a luteinizing hormone.
Pardon me?
LH is luteinizing hormone. Am I saying it right? No.
LH?
Yeah, what is that one?
Luteinizing hormone?
Yeah,
That's
okay.
the initial hormone that maintains the corpus luteum, but that's going to shut down
Right.
because the high levels of estrogen and progesterone shut off FSH and LH production.
Okay, I'm just making sure when you say LH, I'm
Yeah,
remembering
LH is
it correctly.
luteinizing hormone, FSH is follicle.
It's the
stimulating
follicle,
hormone.
yeah, okay.
As I said, you can use those abbreviations. That's on your hormone handout.
Yeah,
All right, and eventually, after about the first trimester, this is going to shut down because now the placenta is the major source of female sex hormones. So that maintains its endometrial line and keeps the pregnancy going. Okay. And
Okay.
then when it does, it kind of shrivels up with scar tissue and becomes corpus albicans. Okay. Make sure you can recognize these, okay, and the cycle, okay, from primordial follicle to primary to secondary, and here's the mature secondary, okay. And then once this releases the egg, that Graafian follicle kind of shrivels up and becomes the corpus luteum. Okay. And eventually, when it does shut down, becomes the corpus albicans.
are these packed?
I think this is supposed to be that, but if I'm asking you to identify it, because it's confusing, it should be yellow, okay?
Gotcha,
I put it here if I want the corpus luteum, I put it here if I want the corpus albicans.
okay,
But know the arrangement, right? And these are primordial, these are primary, that's an early secondary, and here's a late or mature secondary. But as I said before, this will be on the slides, I'm asking you to identify a secondary follicle. I'll make sure it has one that has a antrum. That's the fluid-filled cavity. It's only seen in secondary cavities, okay? Otherwise, you're counting cells. I'm not gonna expect you to do that.
okay,
Because this is actually a secondary. There's more cells around it, and that's the egg cell nucleus and cytoplasm within it. It's called an oocyte.
okay.
Any questions on it? So please learn those two new hormones. Gonadotropin releasing hormone from the hypothalamus to stimulate FSH and LH release. And then human chorionic gonadotropin, HCG for short, which maintains the corpus luteum, which in turn maintains the endometrium by virtue of the progesterone that it produces.
Yep.
Okay. All right, let's look now at what's called cleavage. This is early embryonic development. And cleavage basically is increase in cell number as those cells divide without an appreciable increase in size of the embryo. You can see it on these. And this represents the fertilized egg. One cell. What's this little white thing?
A sperm?
What is it?
Is it? I can't see it, I'm sorry.
It's a little white ball thing. What is it? Sperm cell? No. Sperm is inside the egg. Okay? This is a polar body. This is a polar body. Okay? We're gonna tip your tongue. By the way, let me just go through this once again, a little easier. Gametogenesis. Now, I will tell you right now, you can expect to see this on the lab test without the nice labels. I'll expect you to do the labeling. And specifically, name the cells by name, okay? Don't just say spermatogenic cells. That's fine for the slides, but not here. I want you to know the cell by name, whether it's haploid or diploid, and we're a human cell, how many chromosomes? That's the easy part. Human diploid cells, which are typical, most body cells have how many chromosomes?
No. Diploid is the 40, no. 40, oh my God.
46.
Thank you.
All right. Now, what does diploid mean?
Two pairs.
Chromosomes are paired, okay? It doesn't matter if they're single-stranded or double-stranded. Just if the chromosomes are in pairs, it's diploid. And 2N represents diploid. N means the number of chromosomes. So 2N means chromosomes in pairs. That's diploid.
That's 46.
And most human, yes, be 46 chromosomes in 23 pairs, right? Because the chromosomes are lined up. We have 23 pairs, right? And that last pair are sex chromosomes that determine the sex of the individual. Okay, females, you don't have 2 X chromosomes, and males, 1 X and 1 Y. Now the Y is just a short little stubby. It doesn't look like a Y, it's just a short one. Only seen in males. Okay?
And isn't that what determines the sex?
Does it what?
That's what determines the sex of the baby?
Yes. Yeah, so if your husband, the sick member complains about, why didn't you give me my boy, my girl? Say, hey, buddy,
It's your fault.
it's your fault. You determine the sex, your sperm, because half your sperm are Y-bearing, half are X-bearing. Women's eggs always contain only an X. So it's the guy that determines the sex of the child, not the woman. You might remind them a little biology, they probably won't realize that, but they determine whether they like it or not, right? So, where does this occur? This is called spermatogenesis. Where does it occur?
Ovary?
They'll just say in the male or in the testes. Where specifically in the testes?
Outside the lining, right outside the testicle. That's on the no.
In the walls of the seminiferous tubules, which I showed you on the slides. Okay, the testes are full of these highly folded tubes called seminiferous tubules. It's in the wall that this process occurs, spermatogenesis. You go from a diploid cell, this is the stem cell, it's called a spermatogonium, and again, learn it by name, and it's diploid. 2N means diploid. You can see how many chromosomes in this cell. This is not a human cell. Count them, these are the chromosomes.
Oh, 1, 2, 3, 4, 5, 6.
6, are they paired?
Yes.
Okay, so it's a diploid cell. Okay, now this cell undergoes mitosis to produce another cell just like it, so you don't run out of stem cells. And then this cell is called the primary spermatocyte. You start with a diploid cell, mitosis maintains a chromosome number, so this is a diploid cell too. So up to this point, primary, the spermatogonium, primary spermatocyte, it's all diploid cells. Okay? This cell, primary spermatocyte, undergoes meiosis 1, produces 2 daughter cells. And how many chromosomes in that cell?
3?
Yes,
Yes.
3. Even though double strand doesn't make a difference, they're now single, they're not paired. That's a haploid cell. So this is the secondary spermatocyte. This one's already undergoing division again, all right? Secondary spermatocyte is haploid. And from now on, all the cells here to the sperm are going to be haploid cells. So at the end of meiosis 1, the production of these pair of secondary spermatocytes, haploid cells. These undergo meiosis 2, produce 2 spermatids, also haploid. All you do in meiosis 2 basically is split double-stranded chromosomes
Into single.
You haven't changed the number of chromosomes.
Yep.
And then these spermatids produce the sperm cells. What kind of division are you talking about here?
I don't see a division.
Probably say it's a trick question. It's 1 for 1. There's no division. It's a maturation process. That's called spermiogenesis. 3 things made your humans happen. I went over that in lecture. What are they?
Okay, one more.
It should be obvious.
Development of the tail.
Flagellum, yeah.
Okay, what's another one? Anybody remember?
No.
Shedding excess cytoplasm. This is
And
not to scale. Sperm cells are going to be much smaller than the spermatid. And a third thing is the development of macrosome. Okay? You write that on the board.
mm-hmm.
Okay, so this is called spermiogenesis. This is the last stage of somatogenesis. And you're going from spermatid these cells are all haploid. All right, so three things happen. One is formation of fungilla. Tail. So the sperm can be molded, which it has to be. It's going to be Second is shedding excess cytoplasm. The third thing is the formation of apples. It's a bag of digestive enzymes. This allows the sperm cell to literally digest its way through the barriers. What barriers?
In the zona.
The sperm can physically get into the egg cell cytoplasm to stimulate the egg to finish meiosis II, become a mature egg or ovum. And then when the two nuclei come together, now you got a diploid zygote or fertilized egg. Okay. So it's one for one. One spermatid gives rise to one sperm cell. No division. Just maturation process, those three things. Should you be asked, just in case. All right?
Hint, hint.
All right, this is oogenesis. Where does oogenesis occur? In the female, okay.
Ovary?
In the ovaries, where specifically in the ovaries? Within what? The follicles, right?
Yes.
Follicles are multi-cell structures, and you've got a single egg cell developing within each follicle. All right? So to ask, oogenesis occurs within the follicles in the ovary. Again, we start with the diploid cell. This is the oogonium. It's the stem cell. It's going to produce by mitosis another cell just like it, so you don't run out of stem cells. And then this cell, which is a primary oocyte, also diploid, because all the division here is by mitosis. Mitosis maintains a chromosome number. So if you divide a diploid cell, the daughter cells will also be diploid. Okay, so oogonium, diploid, and primary oocyte, diploid. This cell then undergoes meiosis I and forms two daughter cells. Okay, you can see them here. These are, the big cell is the secondary oocyte, and the small cell, and again, this is not to scale, it's much smaller than this relative to the egg cell, that's called a polar body. And these cells are haploid, just like the secondary spermatocytes, haploid cells. So from here on down, everything is haploid. Now the polar body itself, which is haploid, may divide again into all polar bodies. It doesn't matter because they're all going to degenerate. The important cell is the secondary oocyte. That's the potential egg cell. So the polar body is just a way to get rid of excess chromosomes without wasting a lot of precious cytoplasm in the process. That's why the polar body is extremely tiny. The lion's share of the cytoplasm stays with the secondary oocyte. And what do we see in development of sperm? it sheds a lot of its cytoplasm along with many of its organelles. So be we male or female, all our cell organelles are derived from our mother's egg cell organelles. The sperm really doesn't contribute any except the nucleus and the male chromosomes. Okay, and sometimes there are rare diseases that are associated with the DNA in our mitochondria, and it's all maternal line, because that's where our organelles came from.
Yep.
And mitochondria, if you don't realize, it's got its own DNA.
Yep.
Okay, so this cell then, secondary oocyte, will stop meiosis II, usually in metaphase, and it's an arrested development. It won't continue meiosis II and become a mature ovum or egg cell until a sperm cell enters. And there actually should be a sperm cell in here to make this
Right.
meiosis II complete it and become a mature egg cell. So this is an ovum. That's also haploid. Sperm cell is haploid, you put the two together, and now you've got the diploid fertilized egg or zygote. Okay? Any questions? Learn this.
So fertilized egg is a zygote?
Yes.
Yes, fertilized egg is a zygote. Okay? That applies to other organisms too, even plants, algae, and stuff like this. Okay? Any questions on this? You will see this, I promise you, without the labels. I'll tell you that right now. Learn the cells by name, haploid or diploid, and how many chromosomes were here. 46 if it's diploid, and 23 pairs, 23 single chromosomes. As I said, this is called cleavage, increase in cell number without an appreciable increase in the size of the embryo. And this is going from zygote, the fertilized egg, to the blastocyst. And this occurs in the oviducts, uterine tube. So the early development actually before the embryo even implants. And it's in the oviduct. Does fertilization occur? Up the upper third of the oviduct. And that's why then that zygote will start to develop, it will start to divide by mitosis. You've got a diploid zygote, all the daughter cells are also going to be diploid. So from now on, growth and development, okay, mitosis. So let's look at this. And actually, this is an invertebrate. The early stages are very similar to what we see in urban animals with backbones, okay? This is the fertilized egg. That's the egg cell. So what's this little white thing?
Polar body?
Very good. That's the polar body. Now, this is more to scale. When I said that was not to scale, I meant that. You can see how tiny the polar body is relative to the egg. You don't want to waste a lot of cytoplasm. You're going to keep it with the egg cell, with the organelles, not the polar body. You know this thing may design. And it may hang on for a while, okay? But that's just going to be general. This divides in by mitosis, 2-cell stage, 4-cell stage, 8-cell, 16, 32, 64, 128 by 12, 10, 2048. So it keeps increasing, okay, by powers of 2. And usually it reaches stage, we call it this. In humans, about a 32-cell stage, where the cells now, it's just a solid ball of cells. But notice what's happened. The cells are getting smaller and smaller as they increase in number. This is cleavage. They continue to divide, even smaller, and now they start to pull away from one another, and now you've got a ball of cells with a cavity. This is the blastocyst. The old name, blastula. That's on your handout. Okay? What's important about this stage is, this is the stage it embryos in when it leaves the oviduct, gets in the uterine cavity. This is the stage it embryos in when it implants uterine life and continues to grow. So it's no longer a fertilized egg, it's a multi-cell embryo stage called the blastocyst, a ball of cells in the cavity. Okay. So this is the blastocyst, the old name is called blastula. What's important again? That's the stage it's in when it emerges from the oviduct into the uterine cavity. That's the stage it enters in when it implants uterine life. Increase in size, about the same size, just more cells. You can see the same thing up here, actually a little more awkward. This represents the fertilized egg. That's the nucleus. There's the sperm. And see how tiny that sperm is compared to the egg? When I said sheds excess cytoplasm, What do you mean? See the sperm cell there? Okay. And see, it's inside. Actually, it's really just a vehicle to get those male chromosomes. Okay, so that's the fertilized egg or zygote. And again, 2-cell stage, 4-cell stage, 7-cell, this one would be 8. Keeps going at that 2-cell stage. This is the morula, which means berry. And it sort of looks like what? Maybe a mulberry? Okay, you get the name. Continue to divide, 4 cells. Now you got a cavity. And this is more accurate. This is the inner cell mass. That's the embryo at this point. So it's not only a ball of cells, but it's got a mass of cells on the inside, which are the embryo. That's called the inner cell mass. And that's in your lecture notes, okay? The cells on the outside individually are called blastomeres, because this is the blastocyst. But collectively, they're called the trophoblast. That's what I'd like you to know. Now, the trophoblast is what secretes the HCG. You can see it on your lab objective sheet. This is the source of human chorionic gonadotropin. These cells on the outside of the blastocyst, the trophoblast is what they're called, okay? It's also going to give rise to a membrane, one of the membranes around the embryo, called extrinsic membranes, called the chorion. Talk about soon. Okay. All right, so this is the stage. It's going to implant in your body, and only then does it start to enlarge. Once it's in the metrium, it starts to get bigger. What's going to happen is this inner cell mass is going to flatten out into a bilayered embryonic disc, 2 layers of cells. That's the embryo now. Okay. And again, this is the outside, and then eventually, unfortunately, have a model of this. Look. What happens is a very important process called gastrulation. Up to this point, all the cells are the same in the embryo, okay? You can't tell them apart. I remember a video watching some years ago, I think it was Nova. We got this very distinguished looking British scientist with his starched white lab coat, and he's in his lab and he's saying, there are 3 important events in human life: birth, death, and gastrulation. What's he talking about? Well, for the embryo, this is critical, because here's where you get the differentiation of the cells into what are called the 3 primary germ layers, which you've probably heard of: ectoderm, mesoderm, and endoderm. They're listed here. This is what's going to give rise to all the tissues and organs in the body.
Wow.
Ectoderm will form things on the outside of the body, the more superficial things. The epidermis, the skin. Your sensory receptors, which are associated with the epidermis, as you know. The peripheral and central nervous system, brain and spinal cord, as well as all the nerves, all from ectoderm. By the way, this is in the lecture notes under organogenesis. It wouldn't hurt to learn these. The middle layer is called the mesoderm. This gives rise to things more in the middle of the body, deep to the surface layer. So this would be most of your connective tissue, including the dermis, the skin, your muscles, your skeletal system, bones and cartilage, your excretory organs, your reproductive organs, all come from mesoderm, okay? Then the innermost layer is the endoderm. That gives rise to the GI tract, the liver, pancreas, which are part of the digestive system, as you know, and also the respiratory tract, and a common cavity and tube they share, the pharynx, which is your throat. Okay? Again, these terms are in the lecture notes, and by the way, I have the lecture notes here. If you want to take pictures of it before you leave, you may do so. I'd suggest you learn these, okay? But again, this critical phase, because at this point now you've got differentiation. Now cells are going to form various things. And everything in the body comes from these 3 germ layers: ectoderm, mesoderm, and endoderm. And usually they're shown in different colors. Ectoderm is blue, even though we don't show it here. Mesoderm is red, and endoderm is yellow. Okay. You'll see that in your textbook. And I'll talk a little bit about this when we get back from the break and finish the lectures on this. Okay. All right. I'd like to turn your attention to the extra-embryonic membranes. So these are on your lab objective sheet. There are 4 of these. These are membranes external to the embryo. I want you to know what they are. You will identify them on these. Okay. Also, what they become. What do they give rise to? We're going to start with the outermost membrane, which is this one, shown in blue. This is called the chorion, and it develops from trophoblast. Remember, those are cells on the outside of the blastocyst. So not only do these cells in trophoblast produce HCG, they form the chorion. So this membrane, you can see, outermost membrane around the embryo, it's going to form these little villi, little finger-like projections. Now we saw villi, remember before, the lining of the GI tract. What did they do? What was the purpose of villi?
Increase surface area.
Increase surface area for absorption. Okay, well that's exactly what these things do. They interlock with folds of the endometrium of the mother, the uterus, that will give rise to the placenta. So you can see they're forming here and here as well. So they interlock with folds of the endometrium, and that's the placenta. It's also shown over there. So it starts here, coming from the trophoblast cells, develop the chorionic villi. Eventually, that becomes the embryonic component of the placenta. The internal component would be interlocking folds of the endometrium. Okay, lining of the uterus. So that's the chorion. Second membrane is the amnion. Now normally this is shown in blue, but here it's in orange. This is the amnion. The yellow is the yolk sac. Now at this point, the embryo consists of 2 layers. It's called the bilayered embryonic disc. And it's actually between the yolk sac, which is in yellow, and the amnion, which is in orange. So it's these 2 cell layers here between those 2 membranes. That's the embryo. Amnion in orange and yolk sac in yellow. Okay. Embryo between those 2 layers. Grow around the embryo. You can see it here starting to grow around. So again, it's amnion in orange, and notice it's going to go all the way around and eventually completely envelop the embryo. Now we change colors. This clear bubble here now is the amnion, and the yellow, of course, is developing embryo. That's the yolk sac now. But notice that it's forming a fluid-filled bag, if you will, or sac around the embryo, sometimes called the bag of waters. What happens when a woman's about to give birth?
Or water breaks.
Her water breaks. The amnion ruptures, releases amniotic fluid into the uterine cavity, out the vagina, and hopefully she's where she wants to be, and not in a car rushing to the hospital, okay? That's why sometimes, babies are born
At home.
at home or
In a car.
in the car.
On the highway.
Yeah. And it used to be, in the early days, medieval times, it was probably safer to birth at home than it was by the doctors because they didn't wash their hands. That's why a lot of women died of fevers, puerperal fever, after a successful delivery. But childbirth was very hazardous, and actually the midwives were much better than many of the doctors at the time, because they actually did. Maybe women themselves were aware of the complications, but the men were usually the doctors, and as you know, doctors are a very conservative lot. It's hard to get them to change their ways. It's fortunate now there are a lot more women that are doctors, especially gynecologists. If I were a woman, I want a female gynecologist, not some guy. But anyway, that's just my opinion. All right. As you can see, the embryo continues to grow and expand as that embryo grows. So it's protecting the embryo because now the embryo is actually kind of floating in this fluid-filled cavity. So it protects the embryo from shocks and abrasions. You say, well, how does it breathe? We don't have gills, but we do have gill slits, actually. It's fed what? Oxygen and nutrients via the umbilical cord, which attach the embryo to the placenta. Okay? That's why that's so important. When a baby is born, that it takes a breath. Sometimes the baby's kind of sluggish.
Low
What do you do?
oxygen.
Kind of spot it? Okay. Starts to cry, that's what you want. You want to get that baby breathing. Okay. And then they cut the cord. Okay, and tie it off. And for a while, of course, part of the cord is attached. Eventually, it kind of shrivels up and falls off. Okay, so that's the amnion. Now, the yolk sac, that's in yellow. Now, in embryos that develop outside the body of the mother, the yolk sac is the main source of nutrients. So in a bird egg, like a chicken egg, the water egg, you should be drinking water here, remember? Beverages. In an egg or an embryo that develops outside the body of the mother, like a bird egg, reptilian egg, or even amphibians, that's the source of nutrition, but not in placental mammals. And that's most mammals, except for the egg-laying mammals. Anybody know what they are? Well, you probably heard of, maybe seen them in zoos. We don't have them.
I was just gonna say that platypus?
Yes, the duck-billed platypus. Looks like a duck-bill, strange looking animal. They actually lay eggs. And the other one you're probably not familiar with, that's the spiny anteater. And they're native basically to Australia. Now we also have some mammals that are placental. These are marsupials. They're usually mostly in Australia. If the baby is born in a very immature state, if it crawls along the body of the mother, gets into a pouch of the mother's body, latches onto a nipple, and continues development there. This would be things like kangaroos, wallabies, Tasmanian devils, and so on. Those are marsupial mammals. Okay. But the yolk sac, except in egg-laying mammals, is not a source of nutrition. There's very little yolk. It is actually forms a primitive gut, the GI tract. It's a source of red blood, or it's a source of blood cells and blood vessels. The yolk sac, not a source of nutrition. So again, if you're asked, yolk sac forms a primitive gut or GI tract. It also is a source of blood cells and blood vessels. The yolk sac, not the source of nutrition. So GI tract, the gut, blood vessels.
Can you clarify whether you're asking about mammals or birds?
We're talking
I
about
think he's just gonna be human.
Okay, so I don't have to know about the
You don't need to
birds.
worry about duck-billed platypuses and spiny anteaters, okay? That's just
Wonderful.
FYI if you're interested, okay? I'm a
Yeah.
zoologist by trade, so
Oh,
I know a little bit more than just human. Anyway. Okay, so that's the yolk sac. And you can see it's hanging on to the embryo. Of course, as it develops, it shrinks. Not because it's providing nutrition, but it's becoming incorporated in the gut, source of blood vessels and blood cells. You can see it over there, too, in there. Models, little models there within the wooden case. Okay, the fourth membrane is not even shown in these models. That's called the allantois, which you can see over here. On this. You see stage of development. Here's the section. The pink is the amnion. The yellow is the yolk sac. And the allantois kind of bites off the yolk sac. You can see it here on the right, like a little tail. The amnion gives rise to part of the structure of the umbilical cord, which we see here. That's what ties the embryo to the placenta. You can see part of the placenta there. Okay. And it also gives rise to the urinary bladder, the allantois. And embryos that develop outside the body of the mother, that's a repository for waste products, like a chicken or bird egg, for example, or reptilian egg. So allantois again gives rise to part of the structure of the umbilical cord, which ties the embryo to the placenta, and also helps to form the urinary bladder. So please learn those. Expect to see questions on them. Identify the membrane. What does it become? Good questions. I would like you to know the homologous structures in the male and female reproductive systems. Homologous means have the same embryologic origin. They come from the same tissues. They don't necessarily have the same function. They come from the same place. Analogous would mean same function. So, some are obvious. Testes and ovaries, for example. Penis and clitoris. Scrotum, labia majora, so on. Learn these. Don't be surprised if you see some questions in the lab test. Homologous organs in the male and female reproductive system. Again, homologous means they come from the same tissues, the same embryologic origin. Okay. Any questions? Okay, that's it. So as I said, this will be short.